I want you to imagine, just for a moment, that your own immune system is this elite, highly trained security force.
Right, like fully armed and ready.
Exactly. It's incredibly powerful, capable of neutralizing almost any biological threat you could throw at it. But right now, this elite force is struggling.
They're missing something.
Yeah, the guards are basically walking right past an invisible intruder. It's an enemy that is, disguised itself to look almost indistinguishable from the innocent civilians they're supposed to be protecting.
Which is exactly what cancer does, yeah.
Right. But now consider a completely different scenario. What if doctors could borrow a tiny, almost microscopic sample of that security force?
Just pull a few guards aside.
Exactly. Extract those few guards, drop them into a specialized high-tech training facility, and teach them to spot the precise, unique fingerprint of that invisible intruder.
I love this analogy.
And then, what if, after that training, they could multiply those specific guards by the thousands and send that newly trained army back into your body months before the intruder ever even had the chance to strike again?
I mean, it genuinely sounds like science fiction, but that is the actual frontier of modern oncology we are exploring today.
It really is.
We're looking at a total paradigm shift in how we handle cancer, moving away from just fighting a disease once it's already established this massive defensive foothold.
Right, waiting until it's a huge problem.
Exactly. And instead moving toward proactively arming the body's defenses during a period of, apparent clinical peace.
And that brings us to the real mission of today's deep dive. We are unpacking a truly fascinating 2018 research paper from the journal Oncoimmunology.
Yeah, this is a great one.
It's titled, "A Library-Based Screening Method Identifies Neoantigen Reactive T Cells in Peripheral Blood Prior to Relapse of Ovarian Cancer." That's a mouthful. But our goal here is to figure out the sheer mechanics of how a team of scientists managed to find these hyper-rare cancer-fighting cells floating around in just a tiny vial of a patient's blood.
And the most staggering part of their research really is the timeline.
Yes.
They found these cells months before the patient's cancer actually relapsed on any traditional scan.
Which is just mind-blowing.
Yeah.
So the objective for you listening today is that we're going to break down the highly technical biology behind this breakthrough, but, without watering it down.
No, we're going into full granular detail on the cellular machinery here.
No skimming the hard parts.
Right. We'll explain the foundational concepts the researchers kind of assume their readers already knew, decipher the terminology, and walk step by step through the honestly pretty ingenious methodology they used.
By the end of this conversation, you should walk away with a deep understanding of the mechanics of cutting-edge cancer immunotherapy.
Let's get into it.
So, to set the stage, the paper relies heavily on terms like neoantigens, T cells, and adoptive cell therapy. These are the building blocks of the whole experiment. Let's tackle neoantigens first. Based on my reading, the immune system is constantly scanning the body for antigens, which are basically any protein structure that triggers an immune response.
Right.
But our healthy cells are covered in self-antigens, and our immune system ignores them. So a neoantigen, as I understand it, is what happens when a healthy cell's DNA gets mutated.
Yeah, exactly.
The cell accidentally bakes a brand new mutated protein that has never existed in the body before, effectively waving this microscopic red flag.
You have the core mechanism down perfectly. To build on that, we really have to look at the cellular manufacturing process. DNA is basically a recipe book.
Okay, a recipe book.
Right. When a cell becomes cancerous, that recipe book sustains physical damage, genetic errors.
Like typos in the recipe.
Exactly like typos. If an error occurs in a gene that codes for a protein, the cellular machinery doesn't just stop building, it builds the protein using the corrupted instructions.
Oh, wow. So it just pushes through the mistake.
Yep. And the resulting protein has a completely different sequence of amino acids. It might fold differently or have a different electrical charge.
It looks completely weird to the body.
Right, because it is a completely novel structure. Neo, meaning new, it is distinctly foreign. These neoantigens are the absolute holy grail of modern immunotherapy.
Why are they the holy grail? Specifically?
Because they offer a target that is 100% exclusive to the cancer cell. A therapy targeting a neoantigen won't cause collateral damage to healthy tissue.
But that raises a massive logical question for me. If these neoantigens are such obvious red flags, why does cancer ever successfully grow?
That's the big question, isn't it?
Yeah, why doesn't the immune system just spot this new weird protein and incinerate the cell on day one?
Well, that touches on a phenomenon called central tolerance, combined with the evasive maneuvers of cancer itself.
Okay, let's break down central tolerance.
So, your immune system has a ruthless, fail-safe mechanism designed to prevent autoimmune diseases. When your immune cells, specifically T cells, are first born in the bone marrow, they migrate to an organ called the thymus.
The thymus, okay.
You can think of the thymus as a lethal training camp.
A training camp where they learn what not to attack, right?
Yeah, precisely. In the thymus, specialized cells present the young, immature T cells with practically every normal self-antigen found in the human body.
Showing them pictures of the good guys.
Exactly. It's a biological gauntlet. If a young T cell's receptor happens to bind to one of these normal, healthy proteins, the thymus forces that T cell to undergo apoptosis.
Which is programmed cell death, right?
Right. The cell is literally forced to commit suicide. This ensures that the only T cells allowed to leave the thymus and enter the bloodstream are the ones that are entirely blind to your own healthy tissue.
So the immune system is actively tolerized to the normal self. But because a neoantigen is a brand new mutation that just occurred, yesterday in a lung or an ovary, the thymus never showed it to the T cells.
Right. It wasn't in the training manual.
So the T cells haven't been forced to ignore it.
Exactly. The immune system is completely capable of attacking a neoantigen. But cancer cells are remarkably adept at survival.
They fight back.
Oh, they evolve all sorts of mechanisms to hide those mutated proteins, or they secrete chemicals that paralyze the immune cells that get too close. So the red flag is there and the immune system could see it, but the cancer actively suppresses the response.
Okay, so the specific immune cells doing that spotting and killing are referred to in the paper as CD8 plus T cells.
Yes, the assassins.
I know T cells are a type of white blood cell, but what is the physical mechanism of the CD8 plus part? Why are these the specific cells the researchers care about so much?
Well, T cells are divided into several classes based on the proteins, or co-receptors, on their outer surface. CD8 is one of those specific surface proteins. You can conceptualize CD8 plus T cells as the specialized assassins of the immune system. They don't just coordinate attacks, they execute them.
The actual trigger pullers.
Exactly.
Huh.
When their primary T cell receptor locks onto a specific recognized antigen, say a piece of a flu virus or a cancer neoantigen, the CD8 co-receptor grabs onto the side of the target cell to stabilize the connection.
Like a grappling hook.
Yeah, that's a perfect way to picture it. Once that physical lock is established, the T cell releases granules filled with toxic proteins called perforins and granzymes.
Perforins. Sounds like perforate.
Exactly what they do. The perforins literally punch microscopic holes in the membrane of the cancer cell. Then the granzymes pour through those holes, triggering the cancer cell to dismantle its own DNA and die.
That is brutal.
Highly lethal, but astonishingly specific. A single CD8 plus T cell might spend its entire life circulating the body and never fire its weapons just because it only recognizes one incredibly specific mutated protein that you might never even develop.
Wow. Okay, so we have the highly specific targets, which are the neoantigens, and we have the lethal assassins, the CD8 plus T cells.
Right.
And the overarching treatment strategy the paper is trying to optimize using these two elements is called adoptive cell therapy, or ACT.
Adoptive cell therapy is conceptually brilliant. It acknowledges that the patient's immune system has the right assassins, but perhaps, not enough of them, or maybe they're too exhausted to fight.
So what's the actual logistics of ACT?
It involves abstracting a patient's own T cells, taking them into a highly controlled sterile laboratory, identifying the specific cells that recognize the cancer, and then using growth factors to force those specific cells to divide millions of times.
So you are taking a specialized squad and breeding an entire specialized army.
That's exactly it. You then infuse that massive, hyper-focused army back into the patient's bloodstream.
Okay, but the major logistical bottleneck, and really the entire reason this paper was written, is where doctors actually go into the body to find that initial squad of assassins.
Yes, location is everything here.
The traditional gold standard, mentioned in the research, is hunting inside the tumor itself, looking for tumor-infiltrating lymphocytes, or TILs.
Right, the TILs.
But the authors of this paper aggressively pivot away from the tumor. They want to look in the peripheral blood, specifically peripheral blood mononuclear cells, or PBMCs.
Yes, the blood.
If the war is happening inside the tumor, why would you go looking for soldiers in the blood?
It is entirely about the biological condition of the T cells. Harvesting TILs directly from the tumor has worked reasonably well for certain highly mutated cancers like melanoma, but You are harvesting cells from a literal war
A
zone.
microscopic war zone.
Yeah, and the tumor microenvironment is one of the most hostile places in the human body.
How hostile are we talking? Like, what does that microenvironment actually look like on a chemical level?
It is deeply toxic. Tumors grow so fast that they outstrip their blood supply, leading to hypoxia, which is a severe lack of oxygen.
Okay, no oxygen.
To survive without oxygen, cancer cells switch to a different type of metabolism that produces massive amounts of lactic acid.
So it's acidic, too.
Extremely acidic. So the environment is suffocating and highly acidic. Furthermore, the tumor actively recruits a different class of immune cells called regulatory T cells.
Wait, the tumor uses our own immune cells.
It hijacks them. Regulatory T cells are the immune system's peacekeepers. Their normal job is to stop an immune response after an infection is cleared, just to prevent collateral damage.
Oh, I see.
But the tumor forces them to secrete cytokines chemical signals that essentially drug the CD8 plus T cells, telling them to power down and go to sleep.
That is wild. The environment is acidic, there's no oxygen, and the tumor is actively pumping out chemical sedatives.
Yes. The clinical term for this is T-cell exhaustion. When researchers extract TALs from a tumor, the cells are often biologically beaten down.
They're just exhausted.
They actually have upregulated inhibitory receptors on their surface-like physical exhaustion markers. Even if you take them into a lab and multiply them, you are just multiplying an exhausted, demoralized cell.
That makes total sense. And beyond the biology, there's the surgical reality, right? Yeah.
Exactly. To get TILs, a surgical oncologist has to cut into the patient, remove a chunk of the tumor, and hope there are enough viable immune cells inside it.
Which isn't always possible.
Not at all. If a patient is severely immunocompromised from chemotherapy, or if the tumor is wrapped around a vital organ where surgery is impossible, the TIL approach completely fails.
I was trying to visualize this while reading. Relying on TILs is like trying to recruit soldiers who have been trapped behind enemy lines in a toxic, suffocating swamp for months.
It's a very accurate picture.
They might know the enemy, but they are just too damaged to fight effectively. On the other hand, looking in the peripheral blood is like looking for fresh, naive recruits in a sprawling global population.
Yes.
The blood is a massive renewable reservoir of healthy cells that haven't been subjected to the tumor's acidic swamp. You don't need a scalpel, you just need a standard blood draw.
The peripheral blood offers an incredible advantage in terms of cell quality and patient safety. But, and this is a big, but it introduces a mathematical nightmare.
Because there's so much blood.
Right. The recruits in the blood are fresh, but finding the exact cell equipped to fight a specific neoantigen is astonishingly difficult.
How difficult?
The paper notes that the frequency of a naive neoantigen-specific T cell in the peripheral blood is estimated to be anywhere between 1 in 10,000 to 1 in 10 million.
1 in 10 million. So we are looking for a microscopic needle in a vast biological haystack.
A very large haystack.
And it's not like you can drain all the blood from a sick cancer patient, run it through a filter, and put it back.
Yeah.
You have maybe a few tablespoons of blood to work with.
Exactly.
If the target cells are that incredibly rare, how did the researchers physically find them in such a small volume of liquid?
This is where the sheer mechanical brilliance of the paper shines. They developed a methodology they call the mini-line method.
Mini-lines. Right?
The underlying philosophy was that they couldn't just look for the rare cells, they had to artificially amplify their presence. They had to take a microscopic whisper and turn it into a roar before they could even attempt to listen.
Okay, let's break down the mechanics of this mini-line protocol. How do you amplify a whisper when you don't even know which cell is doing the whispering?
The logistics require immense precision. The researchers started by taking the patient's blood sample and running it through a centrifuge to isolate the white blood cells.
Purifying them.
Specifically purifying the CD8 plus T cells. They ended up with a pool of about 3.3 million CD8 plus T cells.
Okay, 3.3 million assassins.
Now, the traditional approach might be to put all 3.3 million cells into a single flask and test them. But the researchers didn't do that.
What did they do?
They took multi-well plastic plates, imagine a grid of tiny circular indentations, and they systematically distributed the 3.3 million cells across 1,632 individual isolated wells.
Wow, that's a lot of wells.
They calibrated the fluid volume perfectly so that every single well contained exactly 2,000 T cells. Each of these 1,632 wells became its own miniature parallel culture line, a mini line.
Okay, so we have over 1,000 microscopic puddles of liquid, each containing a random assortment of 2,000 assassins.
Right.
What is the mechanism to make them multiply? because they use something called the rapid expansion protocol, or REP. The paper states they use a polyclonal stimulus.
Yes.
I know clonal means identical copies, so does polyclonal mean they are stimulating every single different type of cell at once, regardless of what its target is?
That is the exact definition. They aren't selectively growing the cancer-fighting cells yet. They are blindly forcing every single cell in every single well to divide.
Blindly expanding everything.
To achieve this, they use a highly specific chemical cocktail. The first ingredient is a cytokine called interleukin-2, or IL-2.
What does IL-2 do?
In the human body, IL-2 is the ultimate growth factor for T cells. It binds to receptors on the cell surface and triggers an internal cascade of signals that tells the DNA to start replicating.
Got it.
The second ingredient is an antibody called anti-CD3.
I want to pause on anti-CD3 for a second.
Oh.
How does an antibody force a cell to divide?
It acts as a master biological override. The CD3 protein is physically attached to the T cell's primary receptor. Normally, a T cell only activates when its primary receptor binds to its specific antigen.
Like a key in a lock.
Exactly, but the anti-CD3 antibody is designed to bind directly to the CD3 protein, artificially cross-linking the receptors. It essentially hot wires the cell.
Oh, wow.
It sends a massive go signal into the nucleus, fooling the cell into thinking it has found its target, even though no target is present.
So the anti-CD3 hits the gas pedal, and the IL-2 provides the fuel.
Exactly. And there is a third crucial component here: irradiated feeder cells.
Yeah, this detail fascinated me. Irradiating cells sounds like you were killing them. Why put dead cells in with the T cells you actively want to grow?
Well, they aren't dead. They are just rendered incapable of dividing. The researchers take white blood cells from healthy donors and expose them to a specific dose of gamma radiation. The radiation shatters their DNA, meaning they can never multiply, but their cellular machinery, their metabolism, their protein synthesis, remains completely active for a few weeks.
So they're alive but sterilized, basically.
Yes. T cells are incredibly social cells. If you put them in a plastic well alone with just chemicals, they often die of isolation.
They need friends.
They really do. They require physical, cell-to-cell contact and supplementary signaling molecules to survive the stress of rapid division.
So the feeder cells provide that.
Exactly. The irradiated feeder cells act as a biological scaffolding. They provide the necessary physical contact, secrete supportive survival factors, and then, because they can't divide, they naturally die off and disappear from the culture just as the T cells finish expanding.
That is so elegant. So inside each of the 1,632 wells, the 2,000 T cells are hot-wired by the anti-CD3, fueled by the IL-2, and physically supported by the feeder cells.
Yep. And they sit in incubators for 2 weeks, multiplying blindly. The paper says they expand 1,000 to 5,000 times.
Okay, but this is where I hit a logical wall when I was reading.
Let's hear it.
If you have one cancer-fighting cell and 9,999 cells that fight the common cold, and you multiply everything blindly, you still have the exact same ratio.
Right.
Haven't you just created a louder, much larger haystack? How does this actually help find the needle?
It's a great question. The secret lies entirely in the physical isolation of the tiny wells. We have to look at the mathematics of spatial distribution.
Okay, lay it on me.
Imagine you have that one rare neoantigen-specific T cell hidden among the 3.3 million cells in the initial blood draw.
Yep.
If you put all 3.3 million cells into one giant vat and expanded them 5,000-fold, that one rare cell becomes an army of 5,000 identical clones. But it is now swimming in a vat of 16 billion irrelevant cells.
The ratio hasn't changed at all.
The signal is still completely drowned out by the noise. But with the mini-line method, that one rare cell is pipetted into a single microscopic well along with only 1,999 irrelevant cells.
Ah, I see.
The well is physically sealed off from the rest of the blood. When you apply the rapid expansion protocol to that specific well, the one rare cell becomes an army of 5,000 clones.
And crucially, it's only competing within that well.
Yes. It is now only competing with about 10 million irrelevant cells within that highly confined localized space. You haven't changed the overall ratio of the whole blood draw, but you have drastically shifted the ratio inside that specific well.
You localize the enrichment.
Exactly. You have locally enriched the target to a level where our laboratory instruments can finally detect it.
The analogy that came to mind is a massive, incredibly noisy sports stadium. You are looking for one specific person in a crowd of millions.
A literal nightmare.
Right. Instead of looking in the stadium, you take groups of 2,000 people and put them into a 1,632 separate soundproof rooms. Then you hand every single person a megaphone. That's the rapid expansion protocol.
I like where this is going.
Now, you play the target song. Almost all the rooms stay completely silent. But in the one room that happens to contain the die-hard fan of that song, they scream into their megaphone.
Yes.
Because they're in a smaller room, and because they are amplified, the noise is finally loud enough to penetrate the walls and tell you exactly which room they are in.
That captures the essence of the method perfectly. It isolates and amplifies the signal. And the researchers relied on a deeply fundamental statistical principle here called the Poisson distribution.
The Poisson distribution.
Right, because the target cells are so vanishingly rare in the peripheral blood, like 1 in a million. The laws of probability dictate how they will be distributed when you divide the blood into 1,632 tiny droplets.
Okay.
The Poisson distribution essentially proves that it is mathematically highly improbable for 2 rare cancer-specific T cells to accidentally end up in the exact same well.
Oh, that makes sense.
Therefore, if a well eventually lights up positive for cancer reactivity, there's a 99.3% statistical certainty that all the reactive cells in that well originated from just one single parent T cell that happened to be pipetted into that well on day 1.
It proves the response is clonal. They amplified a single unique fingerprint.
Exactly.
But once they have these 1,632 soundproof rooms full of amplified cells, they have to actually test them. They have to play the song and see who screams.
Yes, the testing phase.
The researchers use an assay called ELISpot. Can we break down the physical mechanics of this test? How do you actually see a microscopic cell reacting to a target?
ELISpot stands for Enzyme-Linked Immunospot. It is a masterpiece of biochemical engineering designed to visualize invisible cellular communication.
How does it work?
Well, when a CD8 plus T cell's receptor successfully binds to its target antigen, the cell becomes activated and immediately begins secreting a cytokine called interferon gamma.
Interferon gamma.
You can think of interferon gamma as the cell's biological battle cry. It's a chemical signal meant to recruit other immune cells to the area.
So they're yelling for backup.
Exactly. The ELISpot test takes place on a specialized plate where the bottom of every well is coated with microscopic capture antibodies. These antibodies are shaped like tiny Ys, and they are specifically designed to bind to interferon gamma.
Okay, they're like little traps.
The researchers take a sample of the expanded T cells from a mini line, place them into the ELISpot well, and drop in the target cancer protein. If the T cells recognize the target, they start pumping out interferon gamma.
And the traps catch it.
As the molecules float down, they get snagged and trapped by the capture antibodies lining the bottom of the well.
So the bottom of the plate is slowly being covered in an invisible layer of trapped battle cries. How do you make it visible?
Through a series of chemical washes. First, the researchers gently wash away the actual T cells, leaving only the trapped interferon gamma molecules behind on the floor of the plate.
Just the invisible signals left over.
Right. Then they add a second liquid containing detection antibodies. These also bind to the interferon gamma, but they have a special enzyme attached to their tail.
Okay, an enzyme.
Finally, they wash the plate again and add a colorless chemical substrate. When the enzyme on the detection antibody comes into contact with the colorless chemical, it triggers a reaction that precipitates a dark, visible pigment.
Wow.
Everywhere a single T cell was sitting and secreting interferon gamma, a distinct colored spot appears on the bottom of the plate. You literally put the plate under a microscope and count the spots. One spot equals one activated cancer-recognizing T cell.
The methodology is an absolute marvel. You divide the blood, you blindly multiply every cell, and then you use the ELISpot to chemically visualize which wells contain these specific assassins.
It's incredible lab work.
But a methodology is just a theory until it is tested on a real biological entity. The researchers needed a test subject with a documented clinical history and a known enemy.
They needed to prove it works in the real world.
Right, prove they could find a needle that actually mattered to a human life.
Which brings us to the centerpiece of the clinical data. Patient 3.
Who was patient 3, and why was her specific type of cancer such a crucial stress test for this entire methodology?
Patient 3 was a woman diagnosed with high-grade serous ovarian cancer, or HGSC. The choice to focus on this specific malignancy is not arbitrary at all.
Why not?
In the world of oncology, different cancers have vastly different mutational landscapes. Cancers that are caused by heavy, continuous exposure to environmental mutagens like melanoma caused by UV radiation or lung cancer caused by cigarette smoke have incredibly chaotic genomes.
They're highly mutated.
Their DNA is riddled with thousands upon thousands of mutations. Because they are highly mutated, they manufacture a massive amount of different neoantigens. They are essentially covered in red flags, making them highly visible to the immune system.
So finding a T cell that reacts to melanoma is comparatively easier because there are just so many targets to choose from.
Considerably easier, yes. But ovarian cancer, specifically HGSC, has a notoriously low to intermediate mutation burden. The DNA is much more stable.
Less typos.
Right. A typical ovarian tumor might only possess a few dozen mutations. The researchers cite a statistic estimating that an ovarian tumor might only have a 39% chance of harboring even one single naturally processed neoantigen that the immune system is capable of recognizing.
That is low, so it hides in plain sight.
It really does. If the mini-line method could successfully find a neoantigen-reactive T cell in a patient with such a quiet tumor, it would prove the assay is sensitive enough to work for almost any solid tumor type.
That makes total sense as a stress test. Let's trace patient 3's timeline, because the temporal aspect when things happened is the most mind-bending part of the paper.
Absolutely. The clinical timeline spans roughly 17 months from her initial surgical intervention.
Okay, walk me through it.
At month 0, she underwent primary debulking surgery. This is a massive abdominal operation where surgeons attempt to physically remove every visible trace of the ovarian tumor.
And they took samples then?
Yes. During the surgery, they collected a sample of the tumor tissue and a vial of her peripheral blood. Following the surgery, she received a standard regimen of systemic chemotherapy to hunt down microscopic disease.
And she went into remission.
For a while, she appeared to be in remission. However, the cancer proved aggressive and resilient. At month 9, she suffered her first clinical recurrence.
How did that manifest?
This manifested as ascites, which is a significant pathological buildup of fluid in her peritoneal cavity, her abdomen.
Ascites fluid isn't just water, right? It's often a direct byproduct of the tumor's presence.
Correct. Ascites is a complex, protein-rich exudate. In the context of advanced ovarian cancer, it is essentially a liquid tumor microenvironment.
A liquid tumor.
Basically, it is heavily populated with free-floating living cancer cells that have shed from the tumor lining the abdominal wall, as well as a massive influx of immune cells attempting to fight them. It's a localized battlefield in a liquid suspension.
And they collected that fluid.
At month 9, doctors drained this ascite fluid to relieve the pressure, and researchers banked the tumor cells floating within it.
Okay. What happened after month 9?
6 months later, at month 15, she suffered a second aggressive recurrence, and they collected tumor cells from her ascites once again. Tragically, the disease ultimately overcame her defenses, and she passed away at month 17.
It's a stark reminder of the stakes involved in this research. But alongside collecting the tumor cells at the recurrences, the researchers were consistently banking her peripheral blood at regular intervals, correct?
That is the critical variable. They drew small vials of blood at pre-surgery, so month 0, and then at 3 months, 6 months, and 12 months post-surgery.
Notice that the 3-month and 6-month blood draws occurred during the period of apparent remission.
Yes, long before the visible clinical recurrence of ascites at month 9.
Okay. To find the neoantigens, the suspects the immune system might be hunting, the researchers needed to sequence the tumor's DNA. The paper details that they performed exome sequencing across the tumor samples taken at the primary surgery, the first recurrence, and the second recurrence. I want to make sure I understand the mechanics of this. They didn't sequence the entire human genome. They only looked at the exome. What is the distinction?
Well, the human genome contains about 3 billion base pairs of DNA. However, only about 1 to 2% of that massive code actually contains the instructions for building proteins.
Just 1 to 2%.
That's it. These protein coding regions are called exons. The remaining 98%, often historically referred to as junk DNA or introns, is involved in structural regulation and timing, but it doesn't get translated into the physical building blocks of the cell.
Okay. Since neoantigens are, by definition, mutated proteins, the researchers didn't waste time or computational power sequencing the non-coding regions.
Exactly. Exome sequencing uses chemical probes to specifically capture and read only the 1–2% of the DNA that codes for proteins.
And what did they find?
By comparing the exome of patient 3's healthy blood cells against the exome of her tumor cells across all 3 time points, they identified exactly 37 non-synonymous somatic mutations.
Let's unpack non-synonymous somatic mutations, because those adjectives carry a lot of weight.
They do.
Somatic means the mutation wasn't inherited from her parents. She wasn't born with it. It occurred spontaneously in a single cell in her ovary during her lifetime, and that cell divided and became the tumor.
Perfectly stated.
And the non-synonymous part is fascinating from an information theory perspective. DNA is read in 3-letter words called codons. Each codon tells the cell to grab a specific amino acid to build a protein.
Right.
Because the genetic code has redundancies, sometimes a mutation changes a letter, a typo occurs, but the new word still codes for the exact same amino acid. The blueprint changed, but the resulting protein is physically identical.
Which would be a synonymous mutation.
Right, and the immune system would never notice it. But a non-synonymous mutation means the typo changes the code so drastically that the cell is forced to substitute a completely different amino acid into the protein chain.
It changes the physical shape, the folding, and the chemical properties of the final structure.
It creates a genuinely foreign object.
Your grasp of the genetics is flawless. They found 37 of these structure-altering typos inside the tumor cells. But, and here's the central biological challenge of the paper, just because a cancer cell possesses a mutant piece of DNA, and just because it builds a mutant protein deep inside its cytoplasm, does not mean the immune system can actually see it.
Why not?
Because T cells do not have X-ray vision. They cannot penetrate the cell membrane to look at the proteins floating around inside. They rely entirely on a spectacular piece of cellular machinery called the Major Histocompatibility Complex class, MHC class I.
MHC class I. I always try to visualize MHC class I as a microscopic storefront window.
A storefront window, I like that.
Yeah, every single cell in your body, with a few odd exceptions like red blood cells, is constantly putting its internal inventory on display in the window, signaling to the outside world, here is a sample of what I am manufacturing today.
Yes.
The CD8 plus T cells act like security guards patrolling the sidewalk, looking into every single storefront window. If a healthy cell is just manufacturing normal, everyday proteins, it puts normal self-peptides in the window. T-cell guard looks at it, recognizes it as normal, and walks right past.
Exactly.
But if a cell becomes cancerous, it starts manufacturing those 37 mutated proteins based on its corrupted DNA. The idea is that it chops up those mutant proteins, puts a mutated piece in the storefront window, and when the T-cell guard looks inside, it instantly recognizes a foreign structure and destroys the entire store.
The storefront window analogy is highly accurate for conveying the function, but the mechanical reality of how the cell prepares that display is even more complex.
Okay, take me into the store. What's happening in the back room?
Inside the cell, there is a barrel-shaped protein complex called the proteasome. You can think of the proteasome as a cellular paper shredder.
A paper shredder.
Right. As proteins reach the end of their lifespan, or if they are misfolded, they are fed into the proteasome, which mechanically chops them up into tiny fragments called peptides, usually spanning about 8 to 11 amino acids in length.
So they get shredded.
Yes. These shredded peptide fragments are then grabbed by a specialized pump called the TATI transporter, which forces them into the endoplasmic reticulum, a cellular factory floor. It is here that the empty MHC class I molecules are waiting.
The empty windows.
Exactly. The critical catch is that the MHC molecule, the storefront window, has a highly specific, rigid, three-dimensional shape. It has a binding groove.
And not everything fits.
Not every shredded peptide will fit into that groove. In fact, the vast majority of peptides simply bounce off. Only the fragments with the exact correct chemical anchors will lock into the MHC molecule.
So it has to be a perfect fit.
Once a peptide successfully locks in, the entire MHC complex is transported to the outer surface of the cell membrane to be displayed to the T cells.
And this is where human genetics makes everything infinitely more complicated. Every human being inherits a different set of MHC genes from their parents. In humans, we call it the HLA system.
All right, HLA.
Just my storefront windows are shaped completely differently than your storefront windows.
100%.
The researchers had a list of 37 mutations found in the tumor's DNA. But they had absolutely no idea which of those 37 mutated proteins would successfully be chopped up by the proteasome, pumped by the TATI transporter, and actually fit perfectly into patient 3's unique, personal MHC storefront windows.
It's a huge guessing game.
If the mutant piece doesn't fit the window, it never gets displayed, and the immune system can never see it.
They faced a massive predictive puzzle. To solve it, they turned to computational biology. They used an advanced predictive software algorithm called NetMHCPan 2.4.
NetMHCPan. What data did they feed it?
They fed the algorithm two datasets. First, they inputted patient 3's specific sequenced HLA type, the exact molecular schematics of her personal storefront windows.
Okay.
Second, they inputted the amino acid sequences of all 37 mutations. The algorithm utilizes artificial neural networks trained on vast databases of known immune interactions.
So it's simulating the fit.
Exactly. It analyzes the electrical charge, the hydrophobicity, and the physical bulk of every possible chopped-up fragment of those 37 mutations and calculates the thermodynamic binding affinity. It mathematically predicts which specific microscopic fragments have the chemical geometry required to lodge tightly into patient 3's MHC groove.
Once the computer gave them the predictions, they had to move from the digital world back to the physical world to test the T cells. How did they do that?
They synthetically manufactured the targets. Based on the algorithm's predictions, they used laboratory synthesizers to literally print out these specific mutated peptides. They grouped these highly predicted targets into what they called minimal pools.
Minimal pools, okay.
However, because even the best algorithms are still just mathematical models and can occasionally guess the proteasome's exact chopping points incorrectly, they added a layer of redundancy.
What kind of redundancy?
They also synthesized longer, overlapping peptides, 15 amino acids long, that covered the entire length of the mutated region of the protein. They called these the 15-mer pools.
Ah, overlapping chunks.
By using overlapping chunks, they essentially guaranteed that the true mutated target was physically present in the test tube, regardless of how the cell naturally chops it. These synthetic peptides, the minimal pools and the 15-mer pools, were the bait they were going to use in the ELISpot assay.
Okay, if we go back to the criminal investigation analogy, the exome sequencing gave the researchers a list of 37 potential suspects. They have their names, but they don't know which of these suspects the immune system detectives, the T cells, are actually actively hunting.
Exactly.
The NetMHCPan algorithm helped them generate the most accurate possible mugshots of these suspects. The synthesized peptides are the mugshots.
That's a great way to think about it.
Now, the researchers are going to take these mugshots, walk into those 1,632 soundproof rooms full of amplified T cells, show them the pictures, and see if anyone recognizes a face and starts releasing interferon gamma.
Yes, the big reveal.
This leads to the massive sifting process. What happens when the amplified immune system is finally confronted with the suspects?
Well, before they could definitively search for the cancer suspects, they had to validate the integrity of their assay.
They needed controls.
Right. They needed to prove that the T cells resting in those 1,632 wells had survived the 2-week rapid expansion protocol, were metabolically healthy, and were physically capable of secreting interferon gamma.
So what do they use for controls?
They ran 2 distinct biological controls. The first involved a peptide known as CEF11.
CEF11 sounds like a barcode. What does it actually represent?
It is an acronym for cytomegalovirus, Epstein-Barr virus, and the influenza virus. CEF11 is a specific, highly immunogenic peptide derived from a combination of these common human viruses.
Oh, common viruses.
Right. The logic here is epidemiological. Almost every adult human walking the earth has been infected by at least one of these viruses during their lifetime.
So we all have memory cells for it.
Exactly. Consequently, our immune systems maintain a circulating population of memory T cells, veteran soldiers who remember the viral face and patrol the blood looking for a recurrence.
And what happened when they tested it?
When the researchers dropped the synthetic CEF11 peptide into the 1,632 mini lines, the ELISpot plate completely lit up. Every single well produced a massive number of spots. This definitively proved that the memory T cells had survived the expansion and were highly reactive.
Okay, that proves the system works for veteran cells that have already fought a war. But neoantigen-specific T cells, by definition, are often naive. They have never seen their target before. Did the researchers run a control to prove they could find a rookie?
They did, and it is arguably the more impressive control. They utilized a peptide called MART1, ARMRT1 is a well-characterized self-protein. It is naturally expressed by normal melanocytes, the pigment-producing cells in your skin, and it is heavily overexpressed in melanoma.
Wait, a self-protein?
Yes. Because it is a normal self-protein, central tolerance in the thymus typically deletes any T cell that binds strongly to it.
Oh, right, the lethal training camp.
Exactly. Any MART1 reactive T cells that manage to escape the thymus and enter the peripheral blood are incredibly rare, functionally naive, and generally have very weak binding affinity.
So they're incredibly hard to find.
Incredibly. The researchers took a subset of 400 mini-lines and challenged them with the MART1 peptide. Remarkably, 23 of those wells showed distinct reactivity.
Wow.
Detecting a naive, low-affinity self-reactive T cell straight out of the peripheral blood is exceedingly difficult. By finding them, the researchers proved beyond a shadow of a doubt that the mini-line method was exquisitely sensitive.
The stage is set. The controls are flawless. The megaphone amplification works for both veterans and rookies. Now comes the main event. They take the pools of the 37 synthetic mutant mugshots and drop them into the 1,632 wells of patient 3's expanded T cells. What did the ELISpot reveal?
Out of the 1,632 mini-lines generated across all 4 of her blood draw time points, exactly 24 wells reacted to the pools of mutant peptides. Just 24 out of 1,632.
That sheer reduction in numbers perfectly visualizes the needle in the haystack problem. But 24 wells reacting to a giant pool of 37 suspects doesn't tell you which specific suspect they are reacting to. It just tells you they hate at least one person in the lineup.
Exactly.
How do you narrow that down to the specific mutation?
They had to systematically deconstrict the response. They took the living cells from those 24 winning wells and subjected them to a second round of the rapid expansion protocol to multiply them even further.
Amplifying the winners again?
Right. Once they had a larger population, they stopped testing them against the entire pool of 37. Instead, they tested each of the 24 lines against the individual isolated mutations one by one.
That makes sense.
Through this rigorous secondary screening, they found that 10 of the lines were either false positives from the initial screen or simply lacked the biological fitness to survive the stress of a second expansion. But 14 lines confirmed true, robust, repeatable reactivity.
Okay, 14 lines.
When they mapped with the specific targets, those 14 lines were reacting to exactly 7 of the 37 specific mutations.
The funnel is getting incredibly tight. We started with 3.3 million cells and 37 genetic typos. We are now down to 14 cell lines and 7 mutations.
Yes.
But reading the results section, the authors state they ultimately ended up with 10 lines, recognizing 5 mutations. How did they lose the others? What was the final purification step?
Well, the researchers were not satisfied with simply having a well that reacted. A single well still contained a mixture of cells. They wanted the absolute purest, most potent monoclonal army of assassins possible for their final testing.
How do you separate the pure assassins from the mix?
To achieve this, they utilized a mesmerizing piece of technology called FACS fluorescence-activated cell sorting.
I know this involves lasers, but the physics of how a laser actually picks up a specific living cell out of a liquid and moves it to a new tube is baffling.
Yeah.
Can you explain the mechanism of FACS?
It is a triumph of fluid dynamics and electromagnetism. First, the researchers needed a way to physically tag the specific T cells that were reacting to the cancer.
Okay, a tag.
When a CD8 plus T cells receptor binds to its target, it goes through activation. As part of that activation, it temporarily pushes a very specific receptor molecule called 4-1BB onto its outer cell membrane.
4-1BB, got it.
The researchers stimulated the 14 cell lines with the target peptides, causing the cancer-fighting cells to push the 4-1BB receptors to their surface. Then they poured in synthetic antibodies engineered to bind exclusively to 4-1BB.
Okay.
Crucially, these synthetic antibodies have a fluorescent fluorophore attached to them, a chemical tag that glows a specific color when hit by a laser.
So the active assassins are essentially wearing microscopic glowing wristbands.
Exactly. Now, the entire liquid suspension of cells is fed into the FACS machine. The machine forces the liquid through a microscopic nozzle, vibrating at thousands of times per second.
Wow, that fast.
Yes. This vibration breaks the stream of liquid into individual microscopic droplets, with each droplet containing exactly one single T cell.
1 cell per drop.
These single-cell droplets fall through a vacuum chamber and pass perfectly in front of a high-powered laser beam.
Okay, the laser.
If the laser hits a cell and nothing happens, the cell falls into a waste collection tube. But if the laser hits a cell and detects the fluorescent glow of the 41BB wristband, the machine's computer registers it instantly.
It sees the wristband.
In a fraction of a millisecond, the machine applies a tiny electrical charge to just that single, specific droplet of water falling through the air. As the charged droplet continues to fall, it passes between two powerful electromagnets. The magnetic field physically deflects the charged droplet midair, pulling it out of the mainstream and dropping it into a completely separate collection tube.
That is unbelievable. It's examining and physically sorting individual living cells midair based on a fluorescent glow, thousands of times a second.
It is profound engineering, but the sorting process is intensely physically stressful on the cells. They are blasted with lasers, electrified, and slammed into tubes.
So some don't survive.
Unfortunately, no. Two of the reactive lines, those targeting genes called KIA 1467 and RRK2, simply did not survive the trauma of the FACS sorting and the subsequent required expansion. They lost viability.
Okay.
So after this incredibly rigorous high-tech gauntlet, the researchers were left with exactly 10 highly enriched pure T-cell lines.
And when they looked at the final targets, what were the 5 mutant suspects that these 10 elite lines were trained to kill?
The 10 lines were strictly specific for mutations occurring in 5 distinct genes: NOX4, CAPN7, ZNF41, or R4C11, and HSDL1.
Okay, those 5.
To be precise on the distribution, 6 of the 10 independent cell lines exclusively recognized the mutation in the HSDL1 gene. The remaining 4 lines each recognize one of the other 4 mutations.
I want to zoom out and look at the magnitude of this filtration process, because the sheer inefficiency of the human immune system is staggering here.
It really is.
The exome sequencing definitively proved that there were 37 unique, non-synonymous mutations physically present in the DNA of the tumor. And yet, after amplifying the blood millions of times, only 5 of those mutations generated any kind of recognizable T-cell response.
Right.
That means 32 unique foreign mutations were completely and utterly ignored by the patient's immune system. Why? Why does the immune system leave so much on the table?
It perfectly illustrates why immunotherapy is so complex. Just because a genetic mutation exists in the DNA does not mean it becomes a viable target. It has to survive a brutal sequence of biological filters.
Walk me through the filters.
First, the corrupted DNA must actually be transcribed into RNA and built into a protein. Second, that mutant protein must be fed into the proteasome. If the proteasome happens to chop the protein right down the middle of the mutation, destroying the unique sequence, the target is lost.
Bad chop, target gone.
Exactly. Third, even if it is chopped perfectly, the resulting peptide might not have the precise chemical anchors required to bind to the patient's specific MHC class I storefront window. It might just wash away.
Doesn't fit the window.
And fourth, even if it miraculously fits into the window, the patient's immune system must possess a circulating T cell with a randomly generated receptor that happens to perfectly match that specific shape.
Right.
If any one of those steps fails, the mutation is biologically invisible. It is a game of molecular roulette, and the odds heavily favor the tumor.
There's another detail buried in the data here that I found crucial. When the researchers were testing these 10 pure lines against the synthetic peptides, they noted a massive difference in a property called avidity.
Ah, yes, avidity.
The T cells reacting to the HSDL1 mutation behave very differently than the T cells reacting to NOX4, CAPN7, ZNF41, and OR4C11. How do we define avidity in this context, and how do they test for it?
Avidity is a critical metric in immunology. It essentially measures the overall functional strength of the binding between the T cell's receptor and the target peptide sitting in the MHC window.
Like a grip strength.
You can think of it as how tightly the T cell grabs onto the target and how sensitive it is to the presence of that target. To test avidity, the researchers set up a titration experiment. They took the synthetic peptides and continuously diluted them, making the concentration of the target weaker and weaker in the ELA spot well.
To see when they stop noticing it.
Exactly.
Yeah.
They wanted to find the absolute minimum amount of peptide required to trigger the T cells to release interferon gamma.
And what were the results?
The results were sharply divided. The T cells reacting to the other 4 mutations, NOX4, CPN7, ZNF41, or 4C11, required peptide concentrations of 10 to 100 nanomolar to trigger a response.
Nanomolar is already a very small measurement. But what about the HSDL1 cells?
The HSDL1 specific T cells were astonishingly sensitive. They continued to release robust amounts of interferon gamma even when the synthetic peptide was diluted down to 10 to 100 picomolar concentrations.
Picomolar, meaning trillionths of a mole per liter. That is a concentration so dilute it is almost conceptual.
Exactly. The HSDL1 cells required 1,000 times less of the synthetic peptide to trigger an attack compared to the others.
1,000 times less.
The HSDL1 cells possessed extremely high avidity. They were hypersensitive tripwires. The other cells possessed low avidity. They needed a massive unnatural flood of the target protein to even notice it was there.
This vast discrepancy in sensitivity becomes a linchpin of the entire paper in the next phase of testing.
It really does.
Which brings us to a massive point of scientific skepticism. I was reading through the methodology, marveling at the chemistry, but a lingering thought kept bothering me.
What was it?
This is brilliant laboratory engineering. They found 10 lines of cells that get incredibly aggressive toward 5 mutated proteins when those proteins are pumped into a plastic well at highly artificial concentration.
Yes.
But a human body is not a plastic Elispot plate. And the real clinical test isn't whether a cell attacks a synthesized chemical puzzle piece. The ultimate question is whether these lab-grown, highly pampered assassin lines can actually look at the real, living, breathing, evasive cancer cells from the patient's body and recognize them as a threat.
Absolutely correct.
Did the researchers actually test the T cells against the physical tumor?
That rigorous skepticism is exactly what drives good science, and it is the exact question the researchers sought to answer in the climax of their experiment. Identifying a reactive T cell against a synthetic peptide in a vacuum is relatively easy. Forcing it to fight the real tumor is the ultimate test.
Let's delve into the mechanics of that ultimate test. How did they execute it?
Recall that during patient 3's clinical timeline, the surgical team collected and froze vials of the ascites fluid from her primary surgery, her first recurrence at month 9, and her second recurrence at month 15.
The liquid tumor samples?
Yes. The researchers thawed these ascite samples. The fluid is a messy mixture of healthy immune cells, dead tissue, and living tumor cells. They needed pure tumor.
How do you build that out?
They utilized microscopic magnetic beads coated with antibodies that specifically target EpCAM, a protein found almost exclusively on the surface of epithelial tumor cells.
Oh, wow.
They poured these magnetic beads into the fluid, the beads stuck to the tumor cells, and they used a powerful magnet to pull the pure, living tumor cells out of the suspension.
Magnetic purification. That is so cool.
It's very efficient. They then seeded these living tumor cells into the Elispot plates right alongside the 10 pure T cell lines.
Okay, so this is the absolute moment of truth. There are no synthetic peptides added to the wells, no artificial boosters. The T cells must physically scan the real tumor cells, natural MHC storefront windows, and see if the target is actually being manufactured and displayed in sufficient quantities to trigger a kill sequence.
Exactly.
And what was the result? Because the paper reveals a massive twist here.
The result was sobering, but illuminating. Out of the 5 specific genetic mutations recognized by the highly trained blood T cells, only 1 was naturally recognized on the living tumor cells.
Out of 5 targets, only 1 actually triggered an attack.
Only 1. The T cell line specific for the HSDL1 mutation aggressively lit up the Elispot plate. They recognized the tumor cells from the primary surgery weakly, but they mounted a massive, overwhelming response to the tumor cells from the first recurrence and a strong intermediate response to the second recurrence.
And the other 4?
The T cell line specifically trained to hunt NOX4, SECIAN7, ZNF41, and OR4C11. They did absolutely nothing. Total silence. When placed in a well with the actual lethal cancer cells that ultimately killed the patient, those highly trained T cells completely ignored them.
I have to use an analogy to process this, because the disconnect is mind-blowing.
Please do.
It is exactly like a high-end security firm training a guard dog. You synthesize the specific chemical scent of a known burglar in a lab, the synthetic peptide. You put that scent on a piece of cloth, and you train the dog to attack it relentlessly. The dog becomes incredibly aggressive and performs perfectly in the training facility.
Good analogy.
But then the actual break-in occurs. The burglar walks right through the living room, and the highly trained guard dog just sits on the rug and watches him steal the television.
It's extremely frustrating, but biologically it makes sense.
Why does a T cell that aggressively attacks the synthetic target completely ignore the real living tumor that harbors that exact same genetic mutation?
The guard dog analogy perfectly frames the biological failures at play. If the dog doesn't bark at the actual burglar, we have to look at the evasive adaptations of the tumor. The first major concept to explore is immune editing.
Immune editing, what is that?
Cancer is not a static, uniform mass of identical cells. It is a highly dynamic population undergoing intense Darwinian evolution inside the human body.
Oh, survival of the fittest.
Exactly. Let's hypothesize about the early stages of the tumor. It is highly probable that the early tumor cells originally expressed one of those other mutations, like NOX4, very clearly. The patient's immune system likely saw the NOX4 protein, recognized it as foreign, and mounted a massive successful attack.
So they were killing it.
The T cells might have killed 99.9% of the cancer cells displaying that NOX4 red flag. But in a tumor of millions of cells, genetic instability means a few cells might have randomly mutated to stop expressing that specific protein.
And those were the ones that survived.
Those invisible NOX4 negative cells survived the immune attack. Because all their competition was killed off, they rapidly multiplied and became the dominant population of the tumor.
Wow.
The immune system essentially applied evolutionary pressure and edited the tumor to become invisible. The T cells circulating in the blood still retain a deep immunological memory of the old target, but the current modern tumor no longer wears it.
Returning to the analogy, the burglar realized the guard dog memorized his cologne, so he simply showered, put on a hazmat suit that blocks the scent entirely, and broke in again. The dog is looking for a scent that is no longer at the crime scene.
Precisely. That is immune evasion by antigen loss. But the researchers dug deeper. They wanted to know the exact mechanical failure for each ignored target. They looked specifically at the OR4C11 gene.
OR4C11, what does that gene normally do?
Normally, the OR4C11 gene codes for an olfactory receptor, a specialized protein expressed in the nasal passages to detect smells. The exome DNA sequencing proved the mutation existed in the tumor's genetic blueprint.
So it's in the DNA.
But the researchers performed a different test on the living ascites tumor cells. RNA sequencing, or cDNA analysis.
Remind me what RNA does again?
RNA is the vital intermediate step. DNA is the master blueprint locked in the nucleus. RNA is the working copy sent to the factory floor to actually build the protein.
Got it.
The RNA sequencing revealed that the tumor cells were not transcribing the OR4C11 mutation into RNA at all. Even though the typo existed in the master blueprint, the cellular machinery was completely ignoring that chapter of the book.
It was a ghost target. No RNA means no protein is ever physically built. If the protein is never built, it can never be chopped up by the proteasome, and it can never be displayed in the storefront window.
Exactly.
The guard dog was rigorously trained to attack a burglar who literally didn't exist in physical space.
That's right.
But what about the other targets? NOX4, CAPN7, ZNF41. Did the RNA sequencing show they were ghosts too?
No, and that makes it even more complex. The RNA sequencing confirmed that those three genes were actively being transcribed. The cell was actively reading the mutated blueprints and manufacturing the corrupted proteins deep inside its cytoplasm.
So why couldn't the T cells see them?
This strongly points to a processing and presentation failure. Cancer cells are constantly mutating their own internal infrastructure. They frequently downregulate or completely mutate the components of the antigen presentation pathway.
Like breaking the shredder.
They break the proteasome so it stops chopping proteins efficiently, or they mutate the TTP transporter so it stops pumping peptides to the storefront window. They sabotage their own supply chain.
So the mutant NOX4 protein physically exists inside the cell, but the cell has disabled the machinery required to display it on the outside. The storefront window is empty.
Yes.
So the burglar is different inside the house, but he is hiding in the basement and systematically avoiding all the windows.
Yeah.
The guard dog patrolling the sidewalk outside has absolutely no line of sight.
It's incredibly frustrating.
This level of evasion is, terrifyingly adaptive. But I want to loop back to that seemingly minor detail you flagged earlier, the avidity.
Yes, the sensitivity.
The researchers noted that the HSDL1 T cells were the only ones that reacted at picomolar concentrations, requiring 1,000 times less peptide than the others. And shockingly, HSDL1 turned out to be the only target actually presented by the real living tumor.
Yep.
Is it just a wild coincidence that the most sensitive cells were the only ones that found the real target?
The authors argue that it is absolutely not a coincidence. It is a profound predictive correlation.
Okay, unpack that.
This raises an existential question for the future viability of adoptive cell therapy. When the patient has a tumor and we identify 50 mutations, how do doctors know which synthetic targets are actually worth pursuing before they waste months of time and millions of dollars growing a T-cell army that might ultimately fail?
Right, because growing them takes time.
The paper suggests that high avidity, that extreme hypersensitivity to vanishingly tiny amounts of peptide, might be a reliable predictive biomarker for true, naturally presented neoantigens.
Oh, I see.
If a T cell requires a massive unnatural flood of 100 nanomolar synthetic peptide in a plastic dish just to activate, it has a structurally weak binding affinity. The reality is that real tumor cells express neoantigens in incredibly scarce amounts.
So it's never enough to trigger them in real life.
A weak binding T cell will never be able to hold onto the rare targets on a real tumor long enough to trigger a kill sequence. But if a T cell reacts to picomolar amounts, it has a vice-like structural grip. It has the binding strength necessary to be a bona fide lethal assassin in the chaotic environment of the human body.
That is a staggering insight for clinical application. HSDL1 was the true target. It was the only scent the burglar was actually wearing when he broke in, and the T cells had the high-avidity noses to successfully track it. The biological mechanisms of how the tumor hid the other targets are fascinating. But the most profound, world-altering finding of this paper isn't just what specific target they found, it is when they found it.
Yes, the timing.
Let's look at patient 3's clinical timeline one last time. This is where the paper essentially builds a time machine.
This is where the laboratory chemistry translates into a vision for the future of patient care. Armed with the knowledge that HSDL1 was the sole, authentic target driving the immune response, the researchers went back to analyze the temporal dynamics of those specific HSDL1 reactive T cells circulating in the patient's blood over the entire 17-month period.
They tracked the exact numbers of the assassins across the 4 blood draws, months 0, 3, 6, and 12.
Yes, and the population dynamics are incredibly telling. At the pre-surgery blood draw month 0 out of the millions of cells they screened through the mini lines, they found exactly one single cell line reacting to HSDL1.
Just one.
The response was barely detectable. At month 3, following surgery and the initiation of chemotherapy, they again found exactly one cell line.
Okay, still very low.
But at month 6, the response violently spiked. They found 5 distinct cell lines robustly reacting to the HSDL1 target in the peripheral blood. The immune system was actively mounting a surge.
5 lines.
And then, at month 12, the researchers screened over 100,000 CD8 plus T cells from the blood and found absolutely zero reactivity. The response had completely vanished from the peripheral circulation.
Before we analyze why they vanished, I want to address a crucial burden of proof.
Okay.
The researchers found these cells in the blood at month 6. But the tumor recurrence, the ascites fluid, didn't clinically appear until month 9.
Right.
How did the researchers definitively prove that the T cells they found floating in the blood at month 6 are the exact same type of T cells that eventually migrated to the abdomen to fight the tumor at month 9? Couldn't they just be 2 different groups of cells that both happen to hate HSDL1?
In biology, you need a barcode to prove identity. For T cells, that barcode is called a clonotype.
A clone?
To understand a clonotype, you have to understand how T cells are created. When a young T cell is developing, it undergoes a chaotic genetic reshuffling process called VDJ recombination.
Sounds complicated.
It purposefully breaks and splices its own DNA to create a completely unique, randomized shape for its primary receptor. Because the reshuffling is completely random, the specific genetic sequence of that receptor acts as a permanent, unique fingerprint for that specific cell and all of its future clones.
Oh, so it's a literal genetic fingerprint.
Yes. The researchers specifically sequenced a hypervariable region of the receptor called the TCRV beta chain. When they sequenced the DNA of the HSDL1 reactive T cells isolated from the blood, they discovered the response wasn't just a single clone.
There's a multiple.
It was a coordinated attack comprised of 3 distinct lineages, or clonotypes. They cataloged their specific V-beta fingerprints, identifying them as V-beta 1, V-beta 5.2, and V-beta 13.6.
So they have the exact genetic barcodes of the assassins in the blood. How do they connect that to the battlefield?
They took the ascites fluid drained during the month 9 recurrence. This fluid is the actual front line where the immune system is actively dying while fighting the tumor. They isolated the exhausted tumor-infiltrating lymphocytes, the TILs, directly from that fluid.
Right.
They stimulated them with the HSDL1 peptide to isolate the active fighters, and then they ran DNA sequencing on their receptors.
And did they match?
The results were a flawless match. The T cells actively fighting inside the tumor ascites at month 9 expressed the exact same 3 V-beta clonotypes, 1, 5.2, and 13.6.
Wow. It's definitive, undeniable proof. The rare cells floating in the blood at month 6 are the exact same lineage as the soldiers fighting and dying in the tumor at month 9.
It is absolute genetic proof. And it forces us to confront the profound clinical significance of the timeline. Patient 3's cancer recurred, aggressively causing fluid buildup and clinical symptoms at month 9.
Right.
But utilizing this mini-line methodology, the researchers successfully isolated, identified, and expanded the exact true tumor-killing T-cell clones from a routine blood draw at 3 months and 6 months.
They found the needle in the haystack 3 to 9 months prior to clinical relapse.
Exactly.
I really want to pause and synthesize the sheer magnitude of what that implies for you listening. If I am tracking the vision here correctly, imagine a patient currently in remission.
Okay.
They have endured the primary surgery. They have endured the rounds of grueling chemotherapy. Their oncologist looks at a high-resolution CT scan or an MRI and sees absolutely nothing. There is no visible tumor.
Clinically, they are clear.
But microscopic residual cancer cells often survive. Using this mini-line method on just a couple of tablespoons of blood drawn during that window of apparent health, doctors could theoretically detect the specific immune cells that are silently fighting that invisible microscopic cancer.
Yes.
They could extract those specific cells, multiply them into an army of billions in a bioreactor, and infuse them back into the patient's bloodstream before the tumor ever has a chance to multiply, adapt, and form that lethalicites fluid.
You have articulated the holy grail of modern oncology. The conceptual framework is called preemptive ACT, preemptive adoptive cell therapy.
Preemptive ACT.
The current model of cancer care is largely reactive. We wait for a patient to relapse, wait for the tumor to grow large enough to be seen on a scan, wait for the patient to become incredibly sick, and then we try to harvest exhausted, suppressed T cells from a massive hospital acidic tumor microenvironment. It is an uphill battle.
And preemptive ACT changes that.
Preemptive ACT flips the paradigm. You strike preemptively, you harvest fresh, healthy, naive T cells from the peripheral blood during the golden window of remission. You expand them to astronomical numbers in a perfect laboratory environment, and you strike the microscopic residual disease with overwhelming, targeted force before it can ever establish a defensive microenvironment to protect itself.
It is a total paradigm shift. But it hinges on timing, which brings up a lingering question about patient 3.
Go ahead.
Why did her T-cell response violently peak at 6 months and then completely, utterly vanish from the blood by 12 months? If those cells were actively fighting the tumor, shouldn't their numbers have stayed elevated to match the threat?
That drop-off highlights the intensely dynamic, almost volatile nature of the human immune response. We are still mapping the rules of this ecosystem.
Do the authors have a theory?
They do. The authors theorize a few biological drivers for this arc. First, the massive spike at 6 months might actually be a delayed reaction to the initial chemotherapy.
How so?
When chemotherapy drugs kill cancer cells, the cells literally burst open, releasing a massive flood of internal antigens, including the HSDL1 mutation, into the bloodstream. This sudden flood of debris acts like a massive vaccine dose, temporarily hyperstimulating the immune system and causing a rapid proliferation of those specific T-cell clones.
Oh, so the chemo caused the spike.
Additionally, chemotherapy is highly toxic to white blood cells. After the treatment ends, the body goes into a state of homeostatic overdrive, rapidly manufacturing new lymphocytes to repopulate the empty blood vessels. This rapid repopulation phase might have temporarily amplified the presence of these rare clones.
The chemo essentially rang the dinner bell and cleared the playing field at the same time. But why the total disappearance by month 12?
By 12 months, the tumor was recurring aggressively. The physical tumor mass was expanding, and the toxic microenvironment was reestablishing itself. It is highly probable that as the tumor grew, it emitted chemical distress signals that drew every available HSDL1 specific T cell out of the peripheral blood circulation and pulled them directly into the peritoneal cavity to fight the localized battle.
The blood reservoir was simply drained empty as all the soldiers rushed to the front line.
Exactly. Alternatively, the massive systemic immune suppression caused by the growing tumor burden might have simply paralyzed and deleted the circulating cells.
That makes sense.
Regardless of the exact mechanism, the takeaway is stark. Timing is everything. There is a specific golden transient window of opportunity during clinical remission where the blood contains these invaluable cells. If you miss the window, the reservoir evaporates.
Wow. We have covered a truly massive amount of deeply complex biology today. We have traced an unbelievable journey.
We really have.
We started with a single vial of blood drawn during a period of deceptive calm. We broke down the staggering logistics of dividing that blood into over 1,000 tiny mini-line wells and using the rapid expansion protocol to force millions of cells to amplify their whispers using the Poisson distribution.
Right.
We walked through the complex computational biology of using exome sequencing and the NetMHC-PAN algorithm to find 37 mutant suspects, and the physical chemistry of the ELISpot assay to visualize the battle cries.
The full sweep.
We saw the harsh reality of immune editing, where the tumor evolves to hide its targets, leaving HSDL1 as the sole high-avidity vulnerability. And finally, we saw how mapping clonotypes unlocks a biological time machine, the ability to identify a tumor-specific immune fingerprint and the assassins trying to kill it, months before the cancer visibly returns on a scan.
It is a masterclass in translational science. The paper elegantly bridges complex bioinformatics, high-throughput cellular biology, and raw clinical oncology into a single, cohesive proof of concept. It conclusively proves that the peripheral blood, when interrogated with sufficient precision, holds the vital blueprint of the body's hidden war against cancer.
Which leaves me with a final, somewhat expansive thought for you to take away.
Let's hear it.
We spent a lot of time discussing how tumors evolve. We saw how patient 3's tumor likely edited itself through Darwinian selection to hide the NOX4 target, or simply stopped reading the RNA for the OR4C11 target.
Yes.
Cancer's constantly moving, constantly mutating to survive the pressures we put on it.
Yes.
But if we now possess the mechanical ability to isolate fresh cancer-fighting T cells from a simple blood draw during remission, could the future of cancer treatment look less like a single definitive battle and more like a continuous rolling software update?
I love that idea.
Imagine a near future where every 3 months you go into the clinic for a simple blood draw. The lab sequences any microscopic circulating tumor DNA, uses the mini-line method to isolate the fresh T cells reacting to the cancer's newest, most recent evasive mutations, expands them into an army of billions, and infuses them back into your bloodstream.
Constantly updating the defenses.
A world where we constantly evolve your immune defenses in real time to stay one step ahead of a mutating disease. It could fundamentally turn cancer from a terminal, acute threat into a perpetually managed, chronic condition.
It is an incredibly provocative and entirely plausible vision of the future of medicine. The intricate tools required to orchestrate that rolling update are being refined in laboratories right now, as this exact paper so beautifully demonstrates.
We want to thank you so much for joining us on this extensive deep dive into the microscopic front lines of immunology. It's dense, it requires patience to understand the mechanisms, but it is the absolute edge of human knowledge. Keep dissecting the science, keep asking the hard questions, and above all, stay insanely curious.
Until next time.
And remember, when the diagnostic landscape seems murky and the traditional tools of medicine seem blind to the threat, sometimes you just need to isolate a few thousand guards in a soundproof room, hand them a megaphone, and truly listen to what they have to say.